All right, guys, welcome back to Underground Science. And in this video, we'll be talking about toll-like receptor 3. Okay, and it's actually going to be for the next few videos. We'll just be talking about a few examples regarding this toll-like receptor. Okay, so let's go and get started. So for this video, we're going to be talking about rotavirus. All right, just as an example, and how toll-like receptor 3 plays a role in this. Okay, so let's say you have an infant that presents with some fever and vomiting and diarrhea, okay, watery diarrhea. And you do the analysis of the stool, right? An analysis of the stool, and you see virus present there, okay, or viral particles. So this example is going to consist of talking about how the rotavirus infects your intestinal epithelial cells and how the toll-like receptor 3 comes into play. All right. So let's go and get started. So let's draw the intestinal lumen. Okay, as we know, the intestinal lumen is made up of epithelial cells. So let's say this is the lumen, and we're going to have our epithelial cells like this. All right. And then we're going to represent the virus in a simple way just by drawing it like that. All right. So this is going to be our virus, our rotavirus specifically, in our intestinal lumen, causing this fever, watery diarrhea, and vomiting. Okay, so let's focus on how that happens. So on our epithelial cells, remember, we talked about, for example, toll-like receptor 5, right? That's present on our epithelial cells also. So just like that, toll-like receptor 3 is also present on our epithelial cells, of course, along with your macrophages um, and your dendritic cells, right? But what we have to focus on is toll-like receptor 5 was on your epithelial cells. So should we draw toll-like receptor 3 on our epithelial cells or inside our epithelial cells, right? So which one's the correct way? So it turns out that toll-like receptor 3 is actually not on the surface of our epithelial cells. It's inside, specifically inside the endosomes. Okay, so what happens is, so we can erase this because this is the wrong representation of toll-like receptor 3. Okay, it's not on the surface, it's inside our cells. So what happens is, let's erase this for now and show what actually happens. So rotavirus, right, contains double-stranded RNA. Okay, so if we drew rotavirus again, like this, it has double-stranded RNA. All right, so double-stranded RNA. So what happens is, first, rotavirus actually binds onto our epithelial cells of the intestines, right? And it binds on via certain proteins. Okay, for example, these proteins are VP4 and VP7. All right, so let's draw that a little nicer, or write that a little nicer. VP4 and VP7, right? So it has these proteins called VP4 and VP7, and it binds to the intestinal epithelial cells. And then endocytosis occurs. And when your intestinal epithelial cells take it in, right, of course, it's going to take it in and form an endosome. All right, so you're going to have rotavirus inside here with its double-stranded RNA. Now remember, we said that drawing the toll-like receptor 3 on the surface is actually wrong because toll-like receptor 3 is not present on the surface like toll-like receptor 5 was. It's actually present inside the endosomes. So remember, we endocytosed the rotavirus, right? So then this is our endosome now. Inside your endosome is toll-like receptor 3. So if we drew it like this would be the correct way to draw it, okay? So this would be your toll-like receptor 3. Now we have our capsid surrounding, okay? So this is our capsid layer that we'll say this white coat right here is the capsid layer, all right? What happens is once this rotavirus is inside the endosome, the capsid layer can actually break a little bit, okay? Instead, so what happens is, right, once inside the endosome, the capsid breaks, or we can say degrades. So it degrades once inside the endosome, And what that does is it exposes the double-stranded RNA, okay? So if the capsid degrades, this exposes the double-stranded RNA that was obviously the genetic material that made up the rotavirus, right? So if we drew that here, let's say the capsid degraded, right? And now the double-stranded RNA is exposed inside the endosome. So then this double-stranded RNA can go ahead and bind to the toll-like receptor 3. Okay, once this happens, we get dimerization, which means two toll-like receptor 3 molecules dimerize. Okay, so once the double-stranded RNA is exposed, it binds toll-like receptor 3, then you get dimerization of toll-like receptor 3. All right. So the toll-like receptor 3 dimerizes. So of course that would mean you have two toll-like receptor 3 proteins that come together and dimerize. Now you can now say that, okay, you have activated toll-like receptor 3. Okay, so once toll-like receptor 3 is activated, remember when we were talking about toll-like receptors 1 and 2 and 5 and 6, right? We said how it eventually activates the MyD88 pathway, right? The difference is toll-like receptor 3 does not activate the MyD88 pathway. Okay, so once toll-like receptor 3 is activated, all right, then what happens is it actually activates the TRIF pathway. Okay, and what the way this is different is because when TRIF gets activated, it then activates IRF3. Okay, now IRF3 is really good at leading to interferon production. Okay, so once IRF3 is activated, you're going to get production of interferon beta. All right. And the reason why this is good is interferon beta actually puts you at an antiviral state. Okay. So this is actually pretty cool. It's a cool way of the immune system, right? Putting you at an antiviral state. Because what can happen is this interferon beta can go to nearby cells and lead to the production of something like protein kinase R. All right. And if nearby cells start producing protein kinase R, what protein kinase R does is it inhibits translation of this virus. Okay. So that's actually a cool thing about interferon beta. Okay. It puts you at an antiviral state. Okay. So we have interferon beta production, right? And another thing that interferon beta does is it actually increases the transcription of MH class 1 genes. Okay, so basically, interferon beta helps upregulate MHC class 1. So what this means is to the cell that's being infected, right? So let's go back up. So this intestinal cell is being infected, right? So when interferon beta starts getting secreted, why? Because of the activation of toll-like receptor 3 and TRIF. And remember, TRIF leads to IRF3 activation, and then activated IRF3 leads to interferon beta production. And we said interferon beta does what? Right? Let's review this. So interferon beta not only does it activate, or not only does it put you in an antiviral state, right, by increasing the production of something called protein kinase R, for example, right? It increases production of protein kinase R, which can then tell nearby cells to inhibit the translation of the virus, right? So that will help stop the spread of the rotavirus for, in this example, right? In case the rotavirus is also infecting other intestinal epithelial cells also. So it's going to help the cells nearby. But another thing interferon beta can do is it can actually, remember on all your nucleated cells of your body, right? You have MHC class 1. Remember, MHC class 1 is supposed to do what? It's supposed to um, help the immune system, right? It's going to help let the CD8 cells, for example, right, know that whether or not the cell itself is infected with a virus or is cancerous or not, right? So, for example, if the cell is infected with a virus, certain viral proteins, right, can get expressed on the MHC class 1. Okay, certain viral proteins can get expressed on the MHC class 1 protein on the cell. And guess who helps upregulate these MHC class 1s? Interferon beta. So that's actually a good thing because then the more so when the viral proteins get expressed on these proteins on MHC class 1 proteins, right? So remember, these are the viral proteins that I'm talking about. In this case, would be um, parts of the rotavirus, right? So when these viral proteins get expressed on the MHC class 1, CD8 cells, right? CD8 positive T cells can actually come in and recognize that, right? CD8 positive T cells can come in and recognize those viral proteins and help destroy this epithelial cell so that the virus can get contained inside that cell and not spread. Okay, so that's the goal of increasing MHC1 expression due to interferon beta, of course. So those are the main functions of interferon beta. All right, so one's going to be to increase MHC1 expression so that you can express more viral particles on it, which will help destroy that infected epithelial cell so you can contain the virus in that cell. And it's also, of course, going to help increase the like certain antiviral particles like protein kinase R so you can stop the spread of the virus. Okay. So that's the importance of interferon beta. But the thing is, right, not only does TRIF activate IRF3, but it also activates something familiar that we've seen in our other toll-like receptor videos, like 1, 2, and 5, and 6, right? It also, TRIF also activates interferon, or not interferon. So we've been talking about interferon for so long, I thought this was interferon. It activates nuclear factor kappa B, right? And we've seen that with MyD88. Remember, for example, toll-like receptor 5, right? MyD88 got activated instead of TRIF, and then MyD88, that pathway activates nuclear factor kappa B. But TRIF, not only does it activate IRF3, it also activates nuclear factor kappa B, and we know that when nuclear factor kappa B is activated, this leads to the production of many different cytokines like IL-1, IL-6, right? IL-12, TNF alpha, right? Many different cytokines. And of course, we know that these cytokines lead to inflammation, of course, right? And that's the more inflammation, the more permeable, the permeability increases, which leads to diarrhea, right? Which is why you see diarrhea. And which is why you see fever, because these cytokines can go to the hypothalamus, right? Which, of course, we know the hypothalamus is important for regulating body temperature. So you can get a fever due to the increased production of cytokines. All right. And that's basically it, right? That I think is good to talk about, right? All these things because it helps us review how toll-like receptor 3 works. And also we went over a good clinical point, right? About a virus, rotavirus, and how that can cause things like fever, watery diarrhea, and so on, right? So I hope the pathway for toll-like receptor 3 made a little bit of sense, and I hope it wasn't too confusing and you guys learned a bit. And I hope to see you guys in my future videos. Thanks for watching.